structure and functions of the plasma membrane. Let's get started. Life depends on plasma membranes. These thin films envelop all cells, separating the cell's interior, or cytoplasm, from the surrounding extracellular environment. But this edge of life is not solid or static. It regulates a steady flow of materials into and out of a cell and performs many crucial functions. Let's begin with the structure of the plasma membrane, whose key components are phospholipids and proteins. Phospholipids form a bilayer in which their nonpolar hydrophobic tails are oriented toward the interior of the membrane, and their hydrophilic phosphate heads face the watery environment on either side of the membrane. Suspended in and attached to this bilayer of phospholipids are various proteins. Biologists use a fluid mosaic model to describe a membrane. The kinky tails of many phospholipids, along with cholesterol found in animal cell membranes, keep the molecules from packing tightly. Thus, the membrane remains fluid, and its components can drift about like partygoers elbowing their way through a crowded room. What about the mosaic part of the model? Just as a mosaic is formed by differently colored and shaped tiles, a membrane is a mosaic of proteins with different shapes and functions. Let's explore some of the activity taking place in and across the plasma membranes of two adjacent cells. Membranes exhibit selective permeability, meaning that they allow some substances to cross more easily than others. Consider oxygen, which enters the cell and is used in cellular respiration, and carbon dioxide, which exits the cell as a waste product. Look again at the plasma membrane, especially its hydrophobic center. Small nonpolar molecules, such as oxygen and carbon dioxide, can easily diffuse across that layer. Even larger nonpolar molecules, such as steroid hormones, can slip through. What substances do you think would have a hard time crossing a membrane? Larger polar molecules, such as glucose, and charged ions, such as sodium, can't cross on their own. They need the help of specific transport proteins to pass through a membrane. Transport proteins may be carrier proteins that shuttle a molecule, such as glucose, across the membrane by changing shape. Other transport proteins provide channels through which specific molecules or ions, such as sodium, can diffuse. Some transport proteins act as pumps, using the energy of ATP to actively move molecules against their concentration gradient. What other types of functions do membrane proteins perform? Some transfer information. Signaling molecules outside a cell may bind to receptor proteins, which transmit the message into the cell. For example, when insulin binds to receptors on a cell, it signals the cell to take up glucose. Attachment proteins provide support by connecting to the cell's cytoskeleton and to the extracellular matrix. These attachments can also relay information about external and internal conditions. Glycoproteins enable cells to recognize each other. The prefix glycol refers to short chains of sugar molecules extending from the protein. Other membrane proteins can recognize these ID tags. The sorting of cells within a developing embryo relies on such cell-to-cell recognition. Junction proteins form long-lasting connections between cells. For example, anchoring junctions hold muscle cells together. Tight junctions create a leak-proof barrier between the cells lining the intestine. And finally, some membrane proteins are enzymes, which may be arranged in clusters that carry out sequential reactions. In addition to its plasma membrane, a eukaryotic cell is packed with membranes enclosing a variety of organelles. Looking at these diagrams of an animal cell and plant cell, what might you conclude about the importance of membranes in the life of a eukaryotic cell? These internal membranes share many of the functions of the plasma membrane. Let's review the functions of the plasma membrane: selective permeability, enzymatic activity, attachment to supporting fibers, signal reception and relay, transport, intercellular joining, and cell-to-cell recognition.